Dr. Ugo Dionne, who did his PhD in the Laval University in Quebec, Canada. And now he's a postdoc in the group of Jean-Claude Gingras in the Lunenfeld-Tanenbaum Research Institute. And I would like to give you the stage.
All right, can you hear me well? Yes, perfect. So hello everyone, I'm happy to have the opportunity to present some of my postdoc work today. I'm interested in how modular signaling is regulated. So as you know, our cells are able to detect different types of signals via plasma membrane receptors, and this will normally trigger the activation of kinases and the assembly of protein-protein interaction complexes. So kinases are really master regulators of the activation of signaling pathways. And many receptors themselves are actually kinases, such as tyrosine kinase receptors. And it's unsurprisingly, these kinases are frequently dysregulated in cancer. So many of our kinases have in their own sequence regulatory regions that are able to modulate the activity of the kinase domain itself. So this allows kinase to only be activated at the right time and place in our cells. So this is frequently dysregulated in cancer via genetic aberrations. One of the most frequent one is point mutations that can be found even in the kinase domain itself, and this can lead to an activated state of the kinase. But in the context of my project, I'm more interested in different type of alterations. For example, kinase can be expressed in cancer cells as truncated form. So these cancer cells can, for example, express a specific splice variant that will lead to the loss of some of the regulatory region of the kinase and an expression of an activated truncated form. Another way is using an alternative start sites that can again lead to the expression of truncated kinases. So these phenomena are pretty rare, but have been found to be able to drive cancer formation of different types of cancers. And some well-known examples are EGFR and ERBB2 truncations. And I am particularly interested in another phenomena, which is the formation of kinase fusions through genomic translocations. So here again, we have a loss of regulatory region of kinases, but we also have a gain of new elements, so coming from another protein fragment that can increase the capacity of the fusions to dimerize and then activate the kinase. So fusions in general are more and more frequently detected. So this is data coming from a recent databases, and now they estimate around 43,000 different fusion proteins have been found in different cancer patients. And based on recurrence, around 2,500 of those could be driving cancer formation because they've been seen in multiple cancer patients. And this is data coming from an older paper, but still very relevant. And it's looking at what type of domains and functional regions are enriched in fusions. And as you can see, kinase domains and other modular domains that are linked with phosphorylation dependent signaling are enriched in fusions. And in fact, there's more than 1,000 fusions that have been found that contain the intact sequence of a kinase domain. And as you can imagine, the vast majority of those have never been studied at the molecular level. So the overall assumption is that a kinase domain is fused to another protein fragment that will contain either disordered region or protein domains that will increase the dimerization potential of the kinase, which will activate it and then stimulate pro-proliferative signaling pathways. And one of the main well-known examples of this is the tyrosine kinase fusion BCR-EBL1 in leukemias. But one more layer of complexity was found recently, so with two seminal papers in 2021, and it's that some tyrosine kinase fusions can form membranous organelles through the biophysical process of liquid-liquid phase separation. So especially this was shown for EML4-ALK in lung cancer. And if we look at cells derived from patients, we can see these puncta here, which are actually signaling platforms that are driven by the fusion and that are sufficient to recruit effectors of the RAS pathway and promote oncogenicity. And this was even more recently shown to be also observed for other types of tyrosine kinase fusions, such as NTRK fusions. And other groups, I found that, in fact, fusions in general are actually very good at forming those phase-separated condensates. And it's even more frequent in fusions that are found in the nucleus and contain transcription factors. So this brings me to the main goal of my project, which is basically to understand better how kinase alterations and kinase fusions are able to hijack our cells. So first question, are these alterations able to frequently form those signaling platforms that are phase separated? How are they able to rewire the cellular signaling machinery and protein interaction networks? So are there a specific mechanism for each fusion or commonalities that could be potentially targeted? What are the actual contribution of the N and C terminal parent? And also, what are the molecular consequences of targeting these fusions with kinase inhibitor to have better understanding of potential resistance mechanism? So as most of you probably know, but when we target kinase oncogenes, it is frequent that there is an emergence of resistance mechanism in patients. So one of the main approach I'm using for my project, it's called BioID, which is a proximity-dependent biotination approach. So for this, you fuse the sequence of a biotin ligase to your protein of interest, which we call a BAIT, which we can express in cells. And when we add biotin to the media, proteins that are in proximity to your protein of interest will be biotinated. And it is estimated that this happens in a radius of around 10 nanometer. So this allows us to then perform a harsh cellular lysis and extract the proteins and then enrich for bytes in the proteins, which we can then identify by mass spectrometry. And by using statistical approaches, we can determine which proteins are enriched in the proximity of our baits compared to negative controls. And this method is important for my project because it is technically difficult to be able to determine which proteins are found in those membraneless organelles because these cannot be extracted from cells. And with BioID, this allows us to biotinate proteins that are found in those condensates directly in living cells. And for my project, I'm looking at the interactions of different kinase alterations, and I'm comparing each of them to the interaction that we detect with the wild type parents. So for a fusion would be an N-terminal and C-terminal parent, for example. So through several collaborations, we were able to assemble a collection of over 50 fusions. Many of those are tyrosine kinase fusions, and they represent pretty well the different kinase domains that are frequently found in cancers. We also have some serine-threonine kinase and some ACH2 domain containing fusions. And importantly, we also have assembled a small collection of around 20 kinase truncations that are found in cancer. So this allows me to compare the molecular mechanism used by the truncation of a kinase versus the fusion of a kinase. So we started this project in kind of a screening approach. So we are using a lentivirus delivery system to express around 50 fusion, 20 truncation, and their wild-type parents in HeLa cells. And then we use Western blot to monitor the expression of our baits and also signaling. And then we use live fluorescence microscopy to determine which of these alterations can form visible condensates. And of course, BioID to look at the protein associations. So our microscopy screen highlighted that many tyrosine kinase fusions are able to form condensate, which is not the case for the other types of alteration that I'm studying. So here, for example, we have cells that have been infected and expressing only GFP as a negative control. And here in comparison, we have EML4 ALK expressing cells. So this is kind of my positive control. This is the fusion that's been shown in 2021 to form these puncta that are signaling platforms. And as you can see, there are different types of kinase fusions that form different types of condensates. And some of them don't form condensates at all. And interestingly, we found that the localization of kinase fusions seem to be determined in great part by the identity of the N-terminal parent. So for example, NPM1 ALK here formed condensates in the cytoplasm, but it's also seen in the nucleolus, and NPM1 is a well-known nucleolar protein. And then with BioID, we're able to determine which proteins are aberrantly recruited to the alterations. So this is a dot plot. It's a small part of the BioID screen. So on the x-axis, we have different baits. So on the left, we have some of the wild-type parents. On the right, some of the kinase fusions. And here on the y-axis, you have different proteins that have been identified as being enriched in one specific or multiple baits. And here, the darker the circle and the bigger the circle mean that this spray was found more abundantly in a specific bait. So I've arranged some of the prey protein depending on their cell signaling functions. And here we can see some classical effectors of phosphotyrosine-dependent signaling. And we can see that these are enriched in kinase fusions and in fact in subsets of kinase fusions. So these would be ALK and MET and NTRK fusions. If we look at PI3K signaling, then this now shifts to PDGFR alpha and PDGFR beta fusions. We also found, interestingly, that many tyrosine kinase fusions are able to recruit component of the JAK-STAT pathway and the interferon response at large. And this is true for many tyrosine kinase fusions, but not for the other types of alteration that I'm studying. And interestingly, we also found that almost every alterations have very specific protein interactions. and they can recruit proteins of very specific signaling pathways. And this seemed to be for fusions to depend on the identity of the N-terminal parent. So we can see here interactions that are found in the wild-type N-terminal parent, and they're also found in fusions, but only in the one that contain the sequence of the N-terminal parent. So this means that many fusions are able to potentially rewire very specific pathways. And some example of this are the PKA signaling, the stripe complex, and the actin remodeling. So in summary of the screen, so we found that many tyrosine kinase fusions are able to form condensate, which is not the case for the other types of alterations, that the localization of the fusion seemed to be driven in big part by the identity of the N-terminal parent, that the classical signaling effectors that are recruited to the fusion seem to depend on the identity of the kinase domain, that many tyrosine kinase fusion can rewire the JAK-STAT pathway, and importantly, that many of the interactions that are found with fusion seem to depend on what is the actual N-terminal parent. So now I'm trying to push forward our understanding of how these alterations work. So I've selected around 20 fusions and 10 truncations that were really strikingly rewiring protein interactions on our screen. And I am now still using our lentivirus system, expressing them in different non-cancerous cell lines that represent a bit better where these alterations are normally found. For example, ALK alterations are frequently found in lung cancer, so now they're expressed in non-cancerous lung cells. And I'm also combining the usage of kinase inhibitors that are already used in the clinic to try to have better understanding of potential resistance mechanism. So we're using Western blot to monitor signaling, microscopy for condensate formation, BioID for interactions, and we're also interested at looking at the overall effect of the expression of these alterations. And so for that, we're using total proteomics and phosphoproteomics. So in the next few slides, I'm just going to show a few examples of the different phenomena we've been observing with these experiments. The first one is regarding targeting fusions with kinase inhibitors that contain the same kinase domain. So here we can see cells expressing NPM1 ALK. As I shown earlier, it's able to form condensates in the cytoplasm, and it's also found in the nucleolus. And when we treat these cells with ceritinib, which is an ALK kinase inhibitor, we see the complete loss of those condensates in the cytoplasm, but we maintain the nucleolar localization. And if we look at some of the BioID interactors, so here on the left, we have DMSO treated, on the right, ceritinib treated, we can see that protein interaction linked with NPM1 are not affected with the treatment, but many phosphodependent signaling effectors are lost or greatly diminished with some exceptions. If we use the same inhibitor to another ALK fusion, so TFG-ALK, which contain the same ALK sequence, we can see that this fusion, for example, is forming multiple condensates in the cytoplasm, but not in the nucleus. And when we treat those cells, we can see that it's the opposite effect that we observe. So we have more condensates and they become bigger. And if we look at the interactions, we can see some similarities. For example, the protein interactions that are linked with the N-terminal parents, so TFG, are not affected. Many phosphodependent signaling proteins are lost, but then we have a new subset of interactions that are actually gained when we do the kinase inhibitor treatment. So this could potentially inform us on potential kinase inhibitor resistance mechanism. So the other observation I wanted to share is regarding different variants of the same fusion. So one good example of this is EML4 ALK. So there are 15 different variants that have been observed of this fusion in lung cancers. So V1, which is the one that's most frequently found, also called the long variant because it contains a longest EML4 sequence, is less linked with resistance to treatment. But the shorter variant called V3, which has a shorter sequence of EML4, is known to be more aggressive and also to potentially resist more to kinase inhibitors. So I've expressed both of these different variants in the non-cancerous lung cells, and I looked at the effect on the total proteome. So by using the new astral mass spectrometer, now it's pretty easy to quantify 10,000 different proteins. And we also enriched for phosphotyrosine peptides using the SH2 superbinder technology in collaboration with Sean Lee. And this allowed us to quantify thousands of phosphotyrosine sites that are modulated depending on the condition. And I'm just going to show you one example here, is if we look at phosphotyrosine peptides in the variant 3 versus the variant 1, we found that there's more than 2,000 different phosphotyrosine peptides that are either only seen in a shorter variant or way stronger in the shorter variant. So this could potentially explain why this variant is more aggressive and more resistant to treatment, as it's able to activate different signaling pathways. So all of this data is kind of highlighting that we think that each kinase alteration or kinase fusion should be seen as a different or distinct oncogenic entity, even if they contain the same kinase domain or if they're a different variant of the same fusion. So this is my last slide. So if we combine our different proteomic approaches, is really giving us a comprehensive view of how these fusions can hijack our cells. So for example, the EML4 ALK, if we look at the proximity of the fusion, we can see interactions that are linked with EML4, other with ALK, and some that are way stronger in the fusion. We can see here with the little p, it means that all of the almost all of the BioD interactors that we see are phosphorylated on phosphotyrosines. Many of them are already known regulatory site of these proteins. And the red arrow here means that these proteins are also overexpressed when we express this fusion. If we go away from the proximity of the fusion and look at the overexpression of other proteins, we found that many oncogenes or proteins that are known with oncogenic progression are overexpressed, and some of them are overexpressed and phosphorylated on tyrosines. And at the phosphoproteome, we have similar findings, so many different proteins already known to be involved in oncogenesis are phosphorylated on tyrosines. So this really gives us a good idea on how this fusion can hijack our cells on multiple levels. So right now, I'm just completing the kinase inhibitor and phosphoproteomic experiments, and we're starting to try to validate some of the finding that we have to try to see which type of proteins or pathways could be potentially targeted to be able to reduce the oncogenicity of kinase fusion and alterations. So I'd like to thank my supervisor, Dr. Anklon Jaga, and my lab mates and all my collaborators for this project. And thank you all for your attention. I would be happy to take any questions.